Welcome, everyone. Couple quick things. We do have a Brightspace quiz today that will start after class. It'll probably start actually about like noon or so. But we do have a Brightspace quiz. Remember, that is going to be due by tomorrow, Thursday at 11:59 p.m. Same format as all the other previous quizzes. So it's going to be worth 15 points, made up of a mixture of some multiple choice questions, some written response questions. This is just some additional practice problems to kind of help you anticipate, prepare for the upcoming exam then as well. We then do have exam number 3 on this Friday. That is going to be covering mostly the catabolism unit, which include things like glycolysis, fermentation, citric acid cycle, respiration, beta oxidation pathways. It is going to be including also the microbial genetics, where we talked about things like DNA, what is a gene, but also things like DNA replication, PCR, gel electrophoresis. It's also genetic transfer. So things like horizontal gene transfer, whether it's transformation, conjugation, or transduction. Biofilms is on there, the lecture set we just finished up last lecture. And then I probably put a little bit also of the kind of a little bit further back, the enzymes and energetics lecture set. I know that was on exam 2. But there will be parts of the enzymes, energetics that's important to understand for catabolism. There might be some from that enzyme and energetics lecture set that's also kind of back on exam 3. Then in particular, things like the energy intermediates, ATP, NADH, proton motive force, because those just also came up again and again and again with the catabolism unit. Okay, so that's going to be the exam coverage. Exam format, again, same as the previous 2 exams, with a mixture of mostly multiple choice or matching questions. But we'll have that section on pathogens of interest. I did post all the pathogens of interest that are on the exam and kind of the slides that we went through all those. Those are posted to Brightspace all in one spot under the study guide materials, and then in the study guide, the study materials for exam number 3 folder. But it does have all of the pathogens of interest that are going to be on this upcoming exam there in one spot if you're using that to study for that part of the exam. There will be a couple short essay questions at the end like there has been on the other exams. Any questions over the exam or exam content? Otherwise, I am just going to kind of keep on going into our next unit where we're going to, again, I've said we're going to start talking mostly about the disease immune system, different kinds of diseases, even some disease treatments. Those are really what we're going to be focusing in on for the remainder of the semester. The exam 4 units always going to be like disease and immunology related. Okay, so with that, we're going to start talking a little bit about disease. And we've talked about it already throughout the semester, especially towards the beginning of the semester. What is disease? What's the definition of disease? Yeah. Yeah, so it's an abnormal condition that affects an organism. So the abnormal means not normal. The organism is not functioning normally, and then it produces noticeable symptoms and signs. What's the difference between a symptom and a sign? Why am I calling those 2 different words? Yeah, subjectivity. Symptoms. What is a symptom? Symptoms are going to be something subjective to the patient, or basically it's something the patient knows they have, subjective to them, but it's not readily apparent to an outside observer. What would be an example of a symptom then? Headache, fatigue, pain. Malaise, that's going to be like fatigue. But again, a big one's going to be pain. Pain is not something that we can readily observe or measure. Typically, we have to ask patients to rate their pain on a scale of 1 to 10. But honestly, different people have different pain tolerance then as well. Some people tolerate pain much better than others. Their score might be different even if they were feeling the same level of pain, for instance. Then. So pain can be very subjective to a patient. Then there are things like signs. And again, signs are more objective changes. Objective things are then going to be things that can be observed, can be measured. And so what might be an example of a sign of disease? Rash, I heard. Fever, you can measure a fever, you can feel that somebody is hot then. It could be lesions, inflammation, and swelling. If something's inflamed, it looks what color? Red. But you can see that, you can observe that. So lesions, swelling, inflammation, fever, things like coughing, sneezing, vomiting, those are things that can be readily seen and observed then. Now, different diseases have different causes, and this is going to come up a lot in this unit. I know I went through this earlier in the semester, but this is going to come up a lot more now. So we're going to revisit some of these. Some diseases are what are called internal dysfunctions. This is where there is an inherent part of the body that becomes defective in some way. So it's basically something about the body goes defective, but it's an inherent part of the body that goes defective, a part of the body that's normal, but then becomes defective in some way. Internal dysfunctions, what might be an example of internal dysfunctions then? Yeah, cancer is probably one of the more readily, probably one of the more classic examples of an internal dysfunction. Cancerous cells are normal body cells that have been changed or transformed, and now they are growing and dividing in an unchecked manner. So what cancerous cells is, they're abnormal body cells that are no longer dividing or growing the way that they're supposed to. We're going to talk about some other examples of internal dysfunctions, but it could be also things like genetic disorders, autoimmune diseases. Those are going to be types of internal dysfunctions. External dysfunctions, this is where something from outside the body gets into the body, and then that is causing the disease. So the disease is caused by some outside or external agent that penetrates into the body. And this is going to typically be when we're talking about things like diseases caused by microorganisms is usually what we're talking about here. So things like flu is going to be an external dysfunction. COVID-19, colds, okay, chickenpox. These are going to be caused a lot of times by bacteria or viruses. Okay, internal dysfunctions, again, I said I was going to list those out a little bit more specifically. Internal dysfunctions include things like genetic disorders, cancer, autoimmune diseases, but also hypersensitivity. By the way, hypersensitivity, this is where we're talking about allergies. Okay, the more technical term for an allergy is a type of hypersensitivity. If I had time, we'd get into it, but there's 4 main types of hypersensitivities. There's type 1, type 2, type 3, and type 4 hypersensitivities. Usually when someone has an allergy, it's type 1. But anyways, we have things like some types of immunodeficiencies are internal dysfunctions. And a lot of times, if something's caused by an environmental factor, it gets lumped into this category. But this is, say, like an allergy due to hay fever. It's not really due to the pollen. The pollen's not really causing the hypersensitivity reaction. It's actually your body's improper response to the harmless pollen. And so the pollen itself is not toxic. It's not causing any damage to your body. It's your body's response to the pollen that's causing the issue. And your body doesn't need to respond. If your body didn't respond to the pollen at all, you would be perfectly fine. So we can classify this a lot of times as a problem with your body, an inherent part of your body, rather than maybe the environmental factor per se. Typically, when we talk about external dysfunctions, we're talking about microorganisms penetrating into the body and causing disease, whether that's a virus, a bacteria, a fungus, or some type of parasitic worm like roundworms or tapeworms, then, for instance. But these are as an outside agent that gets into the body and then causes disease. That's going to be an external dysfunction. Then. We're going to talk more specifically about pathogens in this class. We're going to be mostly talking about then external dysfunctions, externally caused disease in this class, because it is a microbiology class. So with pathogens and disease, we're going to start talking about pathogens. We're going to start talking about how they cause disease, how they spread. This is just some vocabulary. I'm going to start using these terms a lot. Okay, I'm just going to be using these terms fairly commonly throughout the next few lectures. And so these are terminology that you do need to be familiar with. Pathogen, this is a microorganism or an agent that causes disease. A pathogen is something that gets into our body and then causes disease. Agent of disease, this is just a fancy way of saying what is the underlying cause of a disease. For instance, flu disease is caused by the influenza viruses. COVID-19 is a disease, but it is caused by the SARS-CoV-2 virus, and the SARS-CoV-2 virus then is the disease agent, the cause of the disease. Okay? So typically when we're talking about agent, we're talking about the microorganism that causes an external disease, most commonly over the next several lectures. Infection. This is going to refer to the entry of a pathogen into a host body. An infection is how does it get into the host body. Whereas colonization, now that the pathogen has penetrated into the host body, this describes it persisting in the host body and growing up in the host body. So the infection is the initial way it gets in. The colonization is now it's growing up inside the host body and it's causing damage to the host body. Sometimes that colonizes a specific location of the host body. Sometimes it can spread more systemically. We'll talk about that here in a second. Pathogenicity or virulence, I can use those two terms somewhat interchangeably. This is the ability of a pathogen to cause disease, and sometimes there's also a description of how it causes disease or how well it causes disease. The more pathogenic it is, typically the more strongly it causes disease. Okay, virulence is the ability to cause disease, pathogenicity, the ability to cause disease. Some pathogens are only slightly pathogenic, some are very pathogenic. They might spread easily in a population or cause strong disease. And then there are something we're going to talk quite a bit about called virulence factors. Virulence factors is something that a pathogen has. It's an enzyme, a process, a structural feature or structural adaptation. It's something about the pathogen, something it does that allows it to be a pathogen, that allows it to cause disease. So virulence factors, we're going to define this term a little bit more specifically, but it's some type of bacterial trait, bacterial product that allows it to cause disease in a host. So this is, again, I had these all kind of straight up on the slide, but these are some terms that I'm going to be going through and using quite a bit. We're also going to talk a lot in this lecture set about disease cycles and disease transmission. This is a very rough general disease cycle on the slide, where you might start off with a pathogen. But the pathogen, we're always going to start off typically in what is called a reservoir. And a reservoir, this is in some type of environmental source of a pathogen. Where it's at out in the environment before it infects a host. Okay, so this could be, the reservoir could be some type of animals, could be contaminated soil, could be contaminated water, but the reservoir is where the pathogen, the disease agent, is going to be before it infects a particular host. typically, at some point, the host contacts that reservoir. The then pathogen is going to pass from the reservoir into a host. It's going to invade or infect the host. It's going to penetrate into the host, and then it's going to colonize the host. As it colonizes the host, that's persistence and growth within the host. As the pathogen grows up inside the host, though, it's going to cause damage to the host or toxicity to the host. So the pathogen is going to be damaging the host. And then you're going to get some symptoms because of that, some signs and symptoms, and maybe even severe cases, death. But at some point then, that is going to pass from the host back into the reservoir, and then they can repeat that infection disease cycle in a new host. So this is a cyclical set of here of where it's invading a host, growing up inside of the host, passing from a host back out to a reservoir, but then it's going to be passing from the reservoir and infecting a new host. So we're going to start exploring that a little bit more detail. With disease agents, it's not always easy to figure out what is the causative agent of a disease. There is a bunch of just normal microflora, normal bacteria growing on you and in you at any moment. About 25% of your weight is actually bacteria. A lot of them in your gut, in particular, or on your skin. You have as many bacterial cells growing in your body as you do your own cells. Okay? Estimate is you got about 10 trillion cells makes up the normal human adult body on average. So a normal human adult is usually made up of, on average, about 10 trillion of their human cells put together. It's actually estimated you have anywhere from 10 trillion to 30 trillion possibly bacteria in or on your body at any given moment. You're actually outnumbered potentially by bacteria at any given moment in your own body. Okay, so there's a lot. Out of all those normal microflora, though, most of them are either commensalistic. What does commensalistic mean? Doesn't harm us. So commensal relationships are one species benefits and one is neutral in an interaction. A lot of the times, a lot of the bacteria living in and on our body are commensalistic for us. It's good for the bacteria, but they're neutral to us and they cause no harm or no real benefit. But there's a number of these species that are also mutualistic with us, where they benefit from interacting with us, but we also benefit from interacting with them. So a lot of these normal microflora are either mutualistic with us or commensalistic with us. Occasionally, though, we get exposed to a parasitic bacteria. The parasitic bacteria are the ones that cause disease. And if we have disease, how do we determine out of all of that microflora in and on our body, which one is the parasitic one? What's the parasitic species? What's causing the disease? That's really difficult when you're talking about trillions of different bacteria growing on your body, okay? Growing in it. So this, there's a way to identify disease agents, causes disease, and it's called Koch's postulates. It was developed by Robert Koch, who was an early microbiologist. What he did is he came up with a series of steps to identify what is the agent, what is the microorganism species that causes a particular disease. First off, if you got a sick, infected individual, you isolate microorganisms from that infected individual. You then culture those organisms in the lab. But maybe you take a sample from that infected individual, but maybe what you get is like 10 different species. Okay? You grow them on a plate. Do different species have the same colony morphology on the plate? Do different species, do they all form exactly the same looking colonies? No. Something like a Bacillus subtilis growing on a plate looks different than a Staphylococcus aureus, right? Well, what you might get is you might get some colonies on the plate. But maybe there's like 3 different species, say for instance. Well, maybe you infect species 1 into the mouse. Nothing happens. That's not the causative agent of disease. Maybe you affect species 2 into the mouse. Nothing happens. Mouse still healthy. That's not the cause of Asian disease. But then maybe you take colony number 3, you infect that into the mouse, and then maybe in my mouse model, we get disease reproduced. Well, that suggests that this particular species is the cause of Asian disease, but then you should also be able to take a culture from this infected into second infected individual. You should be able to re-isolate species 3 another second time. So this is a series of steps that are conducted to identify what causes the disease. Pretty straightforward. The problem is it doesn't always work. For instance, there's some problems with this. This process works really well when it works, but it doesn't work all the time. One reason it may not work is not all infectious agents are culturable. So some types of bacterial species are non-culturable. We cannot grow them in a lab setting. So for instance, Treponema pallidum, which is a bacteria that causes syphilis, this cannot be grown in lab cultures. It can only be grown inside of host organisms then. And that makes it more difficult to identify as the cause of Asian disease. It takes a longer, much more labor-intensive process. For ethical reasons, also, not all agents can be deliberately reintroduced to host organisms. So, for instance, things like HIV has never been deliberately introduced to humans in a controlled manner to reproduce infections. For ethical reasons, you can't usually do this technique for a lot of human diseases. So, for instance, polio virus would be another example of that then as well. The other thing is not all hosts react the same way to a disease agent. So again, 2/3 of all people are infected with Helicobacter pylori. This causes stomach infections. But out of all those people infected with Helicobacter pylori, only a very small percentage of them are going to end up developing things like stomach ulcers. And so sometimes this Helicobacter is associated with a higher risk of developing stomach ulcers. Doesn't mean you're for sure going to get stomach ulcers, but that can complicate this particular technique. Then there's also many diseases that are never caused by only just one species. So for instance, tooth decay, cavities, that is not just caused by one species at a time. It's usually caused by a mixed species growing in your mouth. All of them are making acids, and then those acids over time gradually result in tooth decay. But it's a consortium of species growing in these biofilms, and often these mixed biofilms made of of 2 or more different species. Those are usually what is causing things like cavities. All right, so we can use Koch's postulates, though, to identify what causes a particular disease. We can also do things like measure how pathogenic a disease agent is. And we can measure pathogenicity through things like infective dose, or sometimes referred to as ID50, or lethal dose, referred to as LD50. The infective dose, this is the number of bacteria, or it could be viruses. It could be just number of a particular pathogens. The number of pathogens that results in an infection in 50% of hosts. So for instance, if you put 50 bacterial pathogens into a mouse, 50% of the time the mouse gets sick. Lethal dose is the dosage or the number of bacteria or pathogens that results in lethality 50% of the time in hosts. And so what we have, and for instance, this is an example of this. In my graph, am I measuring infective dose or lethal dose? I am measuring lethal dose or mortality. So my y-axis on the graph here is percent deaths or percent lethality. So in this particular case, this is a graph that is measuring lethal dose. In this case, maybe I have 2 different pathogens. Maybe I have species A or strain A. Maybe I have species B or strain B. On my x-axis is dosage. in essence, the number of microorganisms, the number of pathogens. So let's say here I infect then a mice population. And maybe I infect the mice with 30 cells of the pathogen. Okay, that's my dosage here. And what I find is on average, if I put 30 pathogens into a mouse, then over here, what I find is 50% of the time that mouse dies if I put a 30 dosage of strain A. So if I infect them with 30 cells of that pathogen, of that strain A pathogen, 50% of the time they die. Okay, that would be my LD50. In this case, again, my LD50 for strain A is 30. Maybe for strain B, if I look at its data though, the LD50 for strain B, that is going to then be a dosage of 50 then instead. So if I infect them with 50 bacterial cells or 50 pathogen cells, then the host dies 50% of the time. Questions about that? Well, if we're looking at this then and we're measuring pathogenicity, which would be considered more pathogenic or more virulent? Is strain A more virulent or is strain B more virulent? We'll go and show of hands here. How many think strain A is the more virulent strain? How many think it's strain B? Strain A is going to be more virulent because it only takes 30 cells to kill 50% of the hosts. That is a stronger pathogen, is more virulent or more pathogenic than strain B, because strain B takes an additional 20 more cells, takes 50 cells to produce the same amount of lethality. Questions about that? You need less of strain A to get 50% of hosts dying. That makes it a stronger pathogen. All right. Some other terms we're going to use. Pathology. This is the scientific study of disease. Pathologists are typically going to figure out what killed a person, for instance. Okay? So pathologists are basically studying disease and what actually resulted in disease. Pathology is often concerned primarily with etiology. What is the actual cause of the disease? Etiology is where did the disease originate? Okay. And then pathogenesis, the manner in which a disease develops within a host, but also spreads from host to host. That is typically what then pathogenesis is describing. When we look at diseases, diseases can be characterized in different ways. Some diseases are communicable. This means the agent of disease can spread from one host to another in the population. So this is a disease that you can catch from someone. What would be an example of communicable diseases? Flu. I think I heard COVID-19. What else? We've talked about several pathogens of interest, for instance, over the course of the semester. What might be some communicable ones? Sometimes pneumonia. Anything that's an STD is going to be a communicable disease. So that could be syphilis, gonorrhea, chlamydia, for instance, some of the ones that we've talked about with this one, monkeypox, HIV. Okay, so there's several different examples of communicable diseases. We're going to talk about some more. Contagious diseases are those that are highly communicable. There are not very many diseases that fall into the contagious category. Contagious diseases spread very, 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 very fast in populations. And typically, contagious disease include things like measles, polio, mumps, and maybe COVID-19. It's on the borderline. COVID-19 is kind of on the border. It's not quite a contagious disease, but it's getting there. Something like flu, not contagious. Okay. For every person that gets the flu, it spreads to about 1.5 more people. Still spreads, but it's not like spreading real fast. By the way, COVID-19 is around 9 to 12. For every person that gets COVID-19, it's estimated they might spread it to about 9 to 12 more people. Okay, measles is up around 16. For every somebody that gets measles, they're going to spread it to around 16 more people. That is kind of the difference between, say, a communicable versus a contagious disease, just to give a little bit of a sense of scale. Non-communicable diseases, these are going to be ones that do not spread in a population. What might be examples of non-communicable diseases? Yeah, cancer. Autoimmune, yep. I also gave the example of like tetanus earlier in the year. But tetanus you get from environmental sources, but it typically does not spread from one person to another to another. Some diseases are acute. This means the disease develops rapidly, but only lasts for a short period of time. What might be an example of an acute disease? Usually it's going to last for a day, a couple days to a week. Yeah. So an example of this is going to be something like flu, for instance. But this is going to be like 2 days, 7 days, let's just say. Maybe 2 to 14 would be this one. Okay. Some diseases are chronic. These are going to be ones that are long-lasting, often then also more slowly developing diseases. What might be an example of a chronic disease? Sorry, yes, there is a chronic kidney disease, and there's a couple different ones of those. Cancer can be. There's different kinds of cancers, which is why I'm hedging a little bit. But there's different kinds of cancers. A lot of cancers are more slowly developing, but there are some that are very quick, unfortunately, and very malignant. So there's different kinds of cancers. Most of them are a little bit more on the chronic side. Alzheimer's would be chronic, yes. Some diseases are going to then be, so these are going to be lasting for months to years, is usually what we're talking about with these diseases, with chronic. Some diseases are subacute. That's kind of an intermediate above. This is going to be weeks to months is the best way to think about this. Subacute diseases kind of last an intermediate amount of time. Mononucleosis can sometimes be an example of a subacute disease. In some cases, C. diff infections can be subacute, which is not very pleasant. Those can last for months in the absence of treatment, for instance. Okay, sometimes. Some diseases are latent. This just means the disease can go dormant or inactive for a period of time. What might be an example of a disease that has a latent stage? Herpes. Not tuberculosis, actually. Tuberculosis just gets gradually worse, but the symptoms are around the entire time. Syphilis has a latent period. HIV can have a latent or dormant period then as well. Some diseases are going to be localized. Localized diseases means they're confined to one particular area of the body. For instance, tuberculosis is confined to what part of the body? lung and just the lungs. So tuberculosis is just going to be in the lungs. That makes it a localized disease. Some diseases are more systemic, though. This means they're going to be spread throughout the body in multiple body locations. And so, for instance, things like septicemias, these are going to be bloodborne diseases. If they're in the circulatory system, they go throughout the body. Okay, and that's bad, usually. Septicemias are very, very bad. Technically, actually, COVID-19 is a systemic disease. People tend to think of it as a respiratory disease. It's actually not a respiratory disease. It is a cardiovascular disease for the most part. What it does is it in particular likes to affect small capillary beds in our bodies. But where do we have a lot of small capillary beds? Lungs. And then also kidney and brain. And you guys ever heard like the loss of taste, loss of smell and stuff like that? That's because the COVID-19 is actually damaging portions of the brain involved in taste and smell senses, and some of the capillary beds in that area. Or at least that's the thought process. They don't have good study on that because, well, there's not a lot of the human brains to look at during it. Anyways. All right. Typically, though, if they're systemic, they're in the blood or the lymph. Have you guys talked about the lymphatic system and anatomy? Okay, a little bit. All right, all right, good. Disease spread. Pathology, epidemiology also studies the incidence and prevalence of diseases in populations. The incidence is a number of hosts developing disease during a particular time period. Okay, this is basically just saying the new cases per a time period. So incidence is focused on newly developing cases of a disease. Prevalence then is going to be both the number of old and new cases over time, but this is going to be total cases of disease is what we're talking about with prevalence. Now, sometimes we have to measure disease spread in incidence. Sometimes we measure diseases and the amount of diseases as prevalence. So for instance, incidence is not a good way to measure non-communicable disease because you don't get a lot of new cases spreading around. Instead, we typically talk about things like autoimmune disorders, cancer cases. We typically talk about them in prevalence, the existing cases. Especially for chronic diseases, we're talking about prevalence. Okay. Typically, though, with communicable diseases or acute diseases, ones that are short-lasting, we're typically more measuring incidence than instead. That's why we got two different terms that we're measuring. Sometimes you need to, especially in the case of acute, short-lasting disease, you're mostly concerned with incidence. For more chronic diseases, you're mostly concerned with prevalence instead, because those are going to be long-lasting cases. Sporadic diseases. These are diseases that only occasionally and at random appear in populations. So there is a random appearance of these diseases. Sporadic diseases, these are ones that pop up, go away. And then they'll not be around for a while. And then suddenly they'll pop up and they'll go away. And it's going to be at random. There seems to be no rhyme or reason for when they come about. Sporadic diseases, an example of this is something like Ebola. Okay, there isn't like a season that you're going to catch Ebola. You're not going to get exposed to Ebola. And in fact, we've gone like a decade with no detectable Ebola cases in human populations before. But all of a sudden, you'll get an outbreak. Okay, a lot of times in Africa, but all of a sudden, you'll just get this outbreak and then it will go away. So that'll be maybe a classic example of a sporadic disease. Endemic diseases, these are diseases that are constantly present in populations. They are endemic or always in the population. What might be an example of an endemic disease? Something that is going to be always circulating in the population, at least at a low level. Colds. A lot of colds are endemic. Flu can be endemic. COVID-19 is currently endemic then as well, for instance. But these are always present, and you could catch them just about any time of the year. Okay, they're always spreading in the population. However, sometimes there are also what are called epidemic diseases. This is where many hosts contract the disease within a short time period. Okay, and most winters we have flu epidemics, for instance, then. We have a flu season, and this is typically then associated with a flu epidemic. Flu is both endemic and epidemic. The terms are not mutually exclusive to one another. Flu is always around, always spreading population, but typically during the winter, you can have flu epidemics where that flu goes from just a low background rate of spread to very quickly spreading through populations. And you get flu season. We can also have things like pandemics. Pandemics, this is a worldwide epidemic disease. And so what's our example of a pandemic? Yeah, so our current example, unfortunately, is COVID-19. But this is rapidly caused a huge number of cases on a global scale in many different countries all over the earth. That is what we're talking about typically with a pandemic disease then. Okay. So we've been talking about some of these pathogenesis terms, but again, something like pathology is looking at causative agents of disease. It's also looking then at spread or transmission of disease when we're talking about pathology. So we're going to start going through these. These are some terms I'm going to start using or defining as we go through disease and disease transmission. Again, this is also terminology that I might expect you to know on, say, exam 4, for instance. Things like host, agent, reservoir, fomites, vector, carrier, nosocomial infections, incubation periods, convalescent periods, decline periods. We're going to have some terminology that you guys can know. We're going to go through these, not now, but we're going to go through these throughout the next lecture. We talk about disease transmission, I'm also sometimes referring to as the chain of infection. And the chain of infection describes the route by which a pathogen moves and is transmitted between one host to another host to another host. So we're talking about communicable diseases here. Okay, when pathogens are transmitted from one host to another host, between hosts. And it's a series of steps that describes the spread of disease. We're going to start off, though, with then looking at this chains of transmission or chain of infection. It is circular. So as it spreads from one host to another host, and then it spreads to another host, and then it spreads to another host. So we're going to start off at reservoirs. And I'm going to talk a little bit more specifically about reservoirs are and how they function in spread of disease. Again, a reservoir, I already defined this a little bit, but an alternative definition, a reservoir, this is some type of living or non-living object that provides a pathogen with conditions for survival, for reproduction, and opportunity for transmission into a host. Typically, this is, again, my other definition that I gave you earlier, some type of environmental source of a pathogen before it gets into a particular host organism. Okay. But it's going to allow that pathogen to survive, maybe reproduce a little, and it's going to allow it to get exposed to a host and infect a host. Now, reservoirs, a lot of different kinds of reservoirs. Different diseases also have different reservoirs. But these reservoirs could be another human. Humans are a very common reservoir of human disease. Again, it's wherever the disease is before it gets into a particular host, one host. But other humans can be a reservoir for disease and spread into one particular human host. Can be animals, could be some type of non-living thing like contaminated water or soil. Again, it's wherever you find the disease agent, the disease pathogen, prior to exposure to a particular host organism. That's what we're talking about with reservoirs. Now, with reservoirs, a main reservoir of disease is other humans. Sick, infected humans will spread disease to other humans. Many people harbor and transmit pathogens to other people. And this is the most common reservoir of human disease, is other humans. This is particularly the case in carriers. A carrier, this is a person that's harboring a pathogen and spreads it, but with no apparent signs or symptoms of illness. So this is someone that has the pathogen in their body, they just don't know it. Okay? But they can still be spreading it around. So individuals that are completely asymptomatic, that have no noticeable symptoms, can still be spreading diseases such as HIV, which causes AIDS and is sexually transmitted, diphtheria, gonorrhea, chlamydia. Again, those are sexually transmitted disease, but often someone who is asymptomatic can be spreading them around. Streptococcal infections, strep throat. Most of the time, people are actually spreading around the Streptococcus pyogenes bacteria, is spreading it to other people before they have any noticeable symptoms or a sore throat themselves. They can spread it down around. Somebody with a strep throat infection can be spreading it for days before they have any noticeable symptoms. Okay? Hepatitis. There's a number of different examples of this then. We can have animal reservoirs. This is where we're typically talking about what are called zoonoses. Zoonoses, these are diseases that are transmitted from one animal species, from some animal species to humans. So say it's a bat disease that spreads into humans. Or we have something like a disease from chimpanzee populations that spread into humans. And by the way, that's what happens with HIV. HIV was originally a chimpanzee disease. Current hypothesis for how it spread into humans is what's called the Hunter hypothesis, that somebody was hunting chimpanzees, basically caught, killed, and infected chimpanzee with HIV. They were butchering it, and some of the chimpanzee blood mixed with a cut on their hand or something like that, and now the HIV virus was in the human, and then it started spreading from that one infected human to others as a sexually transmitted disease over time. Okay, so that makes it a zoonotic disease because we see HIV in chimpanzee populations and also human populations. And we know it started in chimpanzee populations, but spread into humans later. So this is going to be diseases that are transmitted from some type of animal to other things. Includes a lot of different diseases, but it could be things like rabies. How do you get rabies? Bitten by an infected animal, often things like dogs or bats then. Okay, but that's a zoonotic disease. That exposure is through direct contact, consumption of the animal, eating it, or maybe contact with animal waste. For instance, bat guano, you want to avoid that. Bat poop, that is notorious for having diseases in it. Or contaminated animal products, like for instance, leather has happened in some cases. We can also have, though, non-living reservoirs. And this is kind of our last slide for today. Non-living reservoirs, this includes things like contaminated soil, contaminated water, or some type of other inanimate non-living object. For instance, if you have contaminated soil, contact with contaminated soil is how people get things like ringworm, which is not a worm infection. It's actually a fungal disease. I don't know, it's just called ringworm, but it's a fungus disease. There's Clostridium botulinum, Clostridium tetani. Again, these are typically contracted from then contaminated soil. You have waterborne examples where you have contaminated water, because this can cause things like amoebic dysentery, Salmonella typhi, Vibrio cholerae, which causes cholera. These are from drinking contaminated water, then, for instance. And then you get a lot of times these diarrheal dysentery diseases. Questions about that? Or questions about reservoirs? All right. That's a little bit on reservoirs. What we are going to start talking about on Monday, because we have the exam on Friday. On Monday, we're going to start talking about disease transmission and how then diseases spread in populations, including things like contact transmission, vehicle transmission, and vector transmission. But quiz today. Don't forget to take the quiz, and then exam is on Friday.